Today, we're unlocking the science behind what's called the DNA fingerprint.
A single drop of blood, a strand of hair. How do you possibly link that to just one person?
For years, this was a massive headache for investigators. Then, a 1985 breakthrough changed everything.
Sir Alec Jeffreys created what we call DNA fingerprinting, a pattern in your DNA as unique as the one on your fingertip. And you know what's wild? The key isn't our genes, but the DNA sections in between them.
They're called short tandem repeats, or STRs, kind of like little genetic stutters, GATC, GATC, GATC.
So maybe you have 10 repeats of a pattern. I might have 20. That's what makes you, you. Okay, so how do we get a profile? Scientists zero in on 13 specific STR locations, called loci.
First, even a tiny sample gets amplified millions of times using a process called PCR. It's like a genetic photocopier.
So the lab process is simple. Amplify the DNA, separate the fragments, and then analyze the unique pattern.
To separate them, they use a cool technique where an electric current sorts the DNA through a gel.
And what you get is this, a DNA profile. It's a person's unique genetic barcode, right there.
Now, looking at just one spot isn't enough. The real power comes from combining all 13 locations.
The odds of two people matching by chance? Well, it's less than 1 in a quintillion.
To put that in perspective, that's more people than have ever lived. It's unbelievably accurate.
And its uses go way beyond crime scenes, from exonerating the innocent to paternity tests and more.
But a single profile is only useful if you have something to actually compare it against, right?
And that's exactly where CODIS comes in. It's the FBI's massive national DNA database.
CODIS uses that standard set of 13 core loci, making sure all the data is compatible. By 2012, it had millions of profiles and had already aided in hundreds of thousands of investigations.
This is powerful stuff, but it leaves us with a huge question about privacy and our future.